030. Thank you. 30 at 30. That's also a washer setting in the laundry downstairs.
I like the 14-minute one.
Not a fan.
I'm personally a fan of the 8-minute one.
What is that? You just close the door and then open it.
You think about doing laundry.
That's the 0-minute cycle.
For 8
1 minute
minutes.
cycle. Like
You put your clothes into a room and somebody just whispers the word laundry and then that's laundry.
Hey Ed, what's going on quad right now?
Cycle's done.
Pardon me?
Looks fine to me.
No, what's going out?
Oh, sorry. Herc, Argus, and Bubble.
Bubble, okay. We didn't know if it was back row.
What is that on the right?
You guys can look at
Let's
Bubble.
find out, Rennie.
Looks like a tripod fish,
Well, that would be real cool if it was one.
but it doesn't have any tripods deployed.
Place your bets now.
Monopod
Let's
fish.
see.
Monopod fish.
It's just the shadow. There's no fish. It's only shadow.
It used to
Wow,
be
what a
there.
rush. Okay, can you zoom in, please?
Yeah, come
on this
in.
fish that used to be there.
I'll talk.
Oh, that was a pretty good guess, huh?
That's
Yeah.
Well,
It's
Yeah.
tripod,
Ding,
all
ding,
right.
ding, ding.
ding ding.
Wow. Yeah, we gotta check this thing
Great
out.
color on
We are
this one.
currently.
I got
checking
more to
it
go.
Look at all the things it has.
Never
be
seen
funny.
it so
You can
purple
zoom as you
before.
see fit.
That'd be great at Christmas. You could decorate one of these up pretty well.
Mm-hmm.
Coming in some more.
Sure.
It's a really nice color.
I mean, what
Mm-hmm.
is going on?
Yeah.
So are those projections sensory, probably?
Mm-hmm.
Yeah, I'm gonna come up a little bit. Look at those little tabular things.
This is sensing food.
Wow. Cool.
I've never seen them branch like that on the
Yeah.
top.
Beautiful.
Yeah, it's like antlers.
It's beautiful
It's
colors
gorgeous.
and scales, though. Yeah.
Yeah, come out just a little bit.
Huh.
Yeah, the branching is really cool.
Holding.
Yeah, I
And the
haven't
body
seen
color.
that.
Really
Forked. Unusual.
You don't want to bump into one of those things as you float downstream.
Cool. No, not if you're a small critter. Okay, come wide, please.
Coming out.
Okay.
Look at this. It almost looks like a flow that has hardened. And I don't know if that's true or not, but it looks very flowy.
There's another
It's real. Argus is doing that.
same
Yeah.
type.
Go ahead.
Yeah.
Here's another one.
Yep.
Go ahead, George. Maybe. Be with me forever. Oh, George.
So we have an interesting question over here. Someone's asking if elements from the Pacific Garbage Gyre act as an artificial reef or habitat that supports any type of organism.
My understanding is a lot of it is very, very small plastics.
Microplastic.
So it's not like a.
You can't see it.
It's not like a giant raft of plastic you can walk on or anything. It's just these. It's just where little bits end up. But I could be corrected.
Well, there is some biology that makes use of it, hangs out, floats. So yeah, it's a challenge if you start sucking up plastic. How much life are you sucking up too? Plus the plankton that's out there anyway.
I don't think that's an area we've ever gone and studied, is it?
No.
You want to keep stepping, Rennie?
Yeah, I'm gonna go 020 again.
Oh boy.
There we
Mixing
go.
it up.
Bridge nav.
Living large.
One more step. This time bearing 020.
But it's probably an area of research. You know what is living
Roger.
amidst the plastic.
Fish.
Got a fish.
Hey,
It
come
does
back.
not want to be seen.
No,
Nope.
I will not be able to catch him or her.
It's now,
It is now gone.
it is now lore.
Oh, there he
Ah,
comes.
Just kidding.
Bonk.
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 I gotta turn
That was
my.
a disproportionate
It's kind of
fish.
Cuskey-y.
Yeah.
Oh, that is.
Go ahead.
Hello.
Can we zoom?
Yes, that's correct. Thank you.
Interesting.
Zoom in, please.
Oh, there
Yeah,
it
like
is.
Roger, like, yeah, sure.
Nice.
Do you want me to say George next time?
Maybe this guy here.
Yeah, that's correct. Thank you.
That is one big head.
Yeah.
Yep.
Just,
Oopsie.
it's just,
very.
it can't even drive straight because
Okay.
of the size of its
Yeah,
head.
we'll
All right, come
move
by,
on
please.
and stop stunning him with our lights.
I'm gonna learn to drive like him. Just running into stuff all willy-nilly.
Learn. I think you got that down.
Oh, I mean, yes.
Correct. That's not true. I just seize any moment for an insult.
You are a pretty mean-hearted person.
Yeah, I've been told that before.
What was the Roger question?
He wanted to confirm that he called in the right step, which he did.
Oh,
I told him I could say George instead of Roger.
why did you say Roger?
Because like I was like, yeah, yep,
Oh,
Roger.
oh I understand.
Maybe I'll
You
have
know,
that
Roger
for
doesn't mean yes.
Just as like,
It means I understand. I heard you.
yeah,
I understood.
because I said something, he said something, and I was like, Roger.
But it doesn't mean affirmative.
But he already,
If you say go left and he says go right, you can still say Roger. Doesn't mean he's right.
well,
I heard you.
implying I heard it, I would think would be enough.
If you really want to get technical, you could say Roger Wilco.
Roger Wilco.
Or just Wilco. Well, that actually means you're going to do it.
Yeah.
That's not quite right.
Yeah, I don't want that.
That's just affirmative, I guess. Is what you want. Look at this flat top.
Yep,
Top is flat.
might might even go down before it goes up.
Who's
Fish.
to say, really?
Maps.
Oh, a tripod
Maps, I would
fish.
just say.
2 tripod fish. And a little pink thing.
Yeah, a couple of them.
There's a new target in town, and it's this one up here.
Oh, scared him. Yeah, that's way down slope. I'm not going to be able to zoom in on him. Hitting my cow catcher on the back.
All right.
This one swimming.
So who is the Herc pilot on the next watch?
That should be Michael
Mike?
Hannaford.
Okay.
Okay. Are we got to step on?
Yep.
Yep.
Affirmative.
Yeah, maybe we'll get to a less sedimented area. Might be more favorable for corals.
What's our bearing? 2-0, 3-0?
It was 2-0, but I'm gonna change it, I think.
George will love it.
Yeah. Keep changing that bearing. And I'll do another 2-0. Bridge nav. One more step. 30 meters. 020.
Roger.
We're having a lot of fun back there.
Seems like it. So many of them.
We have a lot of fun too, though.
Yeah, we're having fun too. If we, it's just because it's so late that we don't have any friends back here.
Yeah. Do you think Sam will give me another shirt?
I don't know.
Oh yeah, you need one too.
Yeah.
No. I see what's happening here.
Look. Can't. It's not good. Who's that? I think she just held up a $1 bill at us. I don't know what that
That was a
about.
phone.
Oh, okay.
For documentation.
That seems a bit more likely than brandishing money for no reason.
Here's some downslope action.
So can we go back to the question about how do we keep track of the ROVs in the water in relation to the Nautilus?
Haha. We can get back
Won't
to that
escape
question.
it.
Before we get into that, just a I'm waiting for it too, but just wanted to get this out there that if we come across another acorn worm,
Yes.
let's see if we can scoop him.
Ooh.
Scoop it.
Scooping the worm.
Challenge accepted.
Now, can they get away quickly if we were to put it in a bio box? No.
I think they're on the slow side.
They just
Any
kinda-
chance you'd be okay with slurping it?
Slurping seems to be pretty destructive when that's been tried before.
Understood. Scoop. Scoop it is.
Scoop a loop.
Scoop eye.
So yeah, about that ROV navigation.
All right.
Get out pen and paper, kids.
Yeah, you're gonna want
Here
to.
we go.
There'll be a test. Let me call in one more move. How am I gonna keep everyone on their toes? Let's see.
Can't wait. My breath is bated. And I'm
I'm
waiting
gonna
with
go 040.
it. 040I.
Yeah. Well, yeah. I'll do 040. And eventually, we're gonna go more towards that way.
Sure. I'm
Zero.
seeing a little lumpus here.
See a lumpus there?
Yeah.
That
but
could be this lumpus.
could be that lumpus, yeah.
So
That's fine though.
if you're
It's no
here, 04. Maybe just 045 at this point.
045I.
Well, I want to like hit it like right on. See, look.
Yeah, you got it, George.
You got it, George.
You know what's going on.
Okay, George, I see you up there. You can put that move in. Affirmative. All right. Subsurface navigation. So GPS, which we're used to on land via our phones or our cars, that does not work underwater. That's based on time of flight of many different satellites telling you what time it is according to their atomic clocks. And by receiving that information and how long it took to get to you, you can tell where you are in the world because we know where those satellites are and how far away they should be to you. A side note, if you have at least 4 of them, you'll be able to correct whatever clock you have. So your most accurate clock is actually as accurate as the atomic clocks on those satellites, and that is on your phone. So that's GPS, that's what works on the surface of the Earth. But the vehicles we have underwater, and they don't receive those signals. So what we use is a couple different methods of acoustic communications. One, you'll hear it referred to as the USBL, which is an ultra-short baseline, USBL, ultra-short baseline acoustic positioning system. I think the best way to describe this one is to talk about a system we don't use, which is LBL, long baseline system. So with long baseline, there's acoustic transceivers that you position on the seafloor in an array, at least 3 of them. And what they do is they listen for sounds. So you would have a transponder beacon on a vehicle or a diver or something underwater, and it would emit an acoustic pulse, and those that array of transceivers would hear that pulse and know which direction it came from, as well as the range to it, so distance and direction. It could essentially triangulate its position. That's a long baseline. They take a really long time to set up because you have to position those on the seafloor, and we like to go on dives in many different locations over the course of a cruise. Those would be appropriate if you're doing work at the same place over and over again. So what we can use with the ultra short baseline system is you take that an array, at least 3 of these transceivers, and you put them really, 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 really close together, essentially right next to each other, and you mount them in the hull of a ship. And we can detect the phase difference of that same transponder emitting a pulse and still get enough of an idea of the range and bearing to it. So now we have the ship. We know where the ship is according to GPS. We know where the vehicles are in relation to the ship, and we can do a bunch of math and figure out the lat-long positioning of where those vehicles are.
1 sec, is that an acorn worm down there?
Could be.
Let's find out.
Yes, it is.
Can I get front porch on bubble, please?
They're very fragile, easily broken apart, so let's see if we can scoop the entire worm before we try to bring it in.
Okay.
Luckily, this one's laid out in a straight line, not serpentine like the other ones.
We've got a little more movement, but I think we'll be okay to sample. Also, there's not much slope in front of Argus.
After that.
So, but I'll keep an eye on it. Maybe a forward bio box for this guy.
Yep.
Yeah, both of those open.
Yep.
Unless you're feeling spicy, Trevor.
Nope, no spice.
He said no. He said no
Hit
spice.
it with the lasers.
Yeah, you
Massive
bet.
wide.
There's some laser action.
Thank you.
Okay. Let's grab us a scoop and see what we can do. This way? This way. I don't know if it matters. I could do better.
Would you like a zoom on it before we scoop?
Sure, yeah, good idea.
Okay, go ahead, Ed.
Yeah, there you go. Get you out of my light. Thank you. There's the whole organism.
His head's all flat.
Nice.
It's undulating.
Yeah.
That could be the thruster, so.
Yeah.
And come all the way in and get detail shot.
Little fringes.
Is that good back there, Data?
Yeah. Looks
Get those screen caps.
You can tip down a little more.
That's great,
All right.
thank you.
Okay.
Coming out. Do you have something, Avery?
Nope, you got it. Probably 15, 20 more degrees.
Massive
Oh, I know it's
wide.
been going in.
You can keep tipping down. You got quite a bit more still.
You want me to come in a bit?
Yeah, please.
Yeah, coming in.
Good there.
Brighten you up a little bit too. Hey, little buddy.
Sediment is cooperating.
We had a chat ahead of time.
Yeah.
Yeah, me and the sediment did.
Great job.
It's not sampled yet. Don't jinx it.
Stay in there, boy.
Okay, come wide, please, Ed.
Wide eye.
Okay, Avery, can you please open my tool tray? All the way.
Wow.
Oh, the porch light.
Good there.
How I missed you.
I know, you don't usually say that, but in this case, it's perfect. I'm gonna put this one on the port side. I don't like this stance at all. I'm gonna do this.
Out you go.
Come on.
Should just slide right out now. That's some gummy sediment.
There it is. It flung out.
It flung out.
Yeah.
Sample collected.
Okay, please tool tray in.
Wow. Yeah, I'll hit the I believe button. It's down there. Nice job.
That was sample 003. Is that correct?
That is correct.
Okay. I'll wait for you to catch up before I do anything.
Stow the scoop and stuff too.
Scoop stow.
Scoop stow. Jack's scoop stow.
Oh, wow.
That was physically painful.
That one hurt a little, but it also felt good too.
Thanks, Randy. That's all I can ask for from you.
Jacques.
Was it a
Scoop
joke?
stow.
I missed a
Jacques
joke?
Scoop stow.
Oh, no. No.
Well, he did it.
I certainly did.
No.
Can't be taken
Why
back.
are you still holding it?
You have a
Sam,
wrist roll
we
going on.
we gotta get going here.
We scoop it and go.
Scoop and go.
Scoop and run.
Scoop and run.
That wrist rotate thing is back, Trevor?
You betcha. That's just because I haven't called it today.
Huh.
Or,
Okay.
you know, this minute. Okay. Let's see there.
Makes for very exciting sample collecting. We can
What's
scoop
that?
something at any moment.
Yeah.
It could self-sample at any moment.
Ready? 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 Oh, scooping.
They should really
All right.
make a first-person ROV
There's a cool
pilot
tunicate.
game where you always have the
scoop, yeah.
You always have yeah. Well, you can change your weapon, but scoop is one of them.
Doot, doot,
That's
doot,
good.
doot,
Good thought,
doot,
yeah.
doot.
Yeah. Magnetico.
Ben's gonna make it.
Oh no.
Oh, no.
Oh no. Oh no.
What have you done?
Magnet, work with me. There we go.
There you go.
Nice. Bong. Okay. Let's see, where is
Okay.
Argus now, and where is the target? Okay, I'm pretty confident that we're gonna go 055. Let me know when you're happy.
I'm happy.
Okay,
If I were a clam,
bridge nav.
I'd be happy.
One step, 30 meters, 055. Thank you.
Yeah. Come on.
There is that wall at 055.
Roger. We're going 045?
055.
Be interesting to see how our science team collects that specimen out of the box.
How they do it?
Yeah.
Yeah.
Very carefully.
Would
Maybe
you use a
scoop
scoop?
it,
Yeah.
or maybe just put the container into the water and try and get it in there.
We'll see how it returns to the surface.
Intact.
Angrily.
Stock tunicate.
Mm-hmm.
Never seen so many of those. Ooh, Umbellula.
Yeah.
Umbellula.
And something cute to the left of it, too.
Well,
Stock
it's
something.
cute too.
Oh, actually, that's not an Umbellula.
That's
That's
not an
a
Umbellula.
mushroom coral.
Mushroom coral,
Yeah.
yeah.
That's.
Can you please zoom in on the mushroom coral?
Anthomastus.
Yeah. Was that a...
Yeah, zoom in now, please.
Okay, thank you.
Cool.
What type of animal is this?
Anthomastus mushroom coral.
Thank you.
It's because when it retracts, it looks like a mushroom.
Yep. Type of octocoral. If you count the little tentacles there should be 8.
Read my mind. I was just counting the...
And what was behind it here? This other cute thing?
Crinoid, yeah.
Crinoid. Okay.
Oh,
Cool.
and another
Couple
one.
more.
Crinoid
yeah.
on the rocks, too.
Crinoid on the rocks.
Prefer mine neat.
Oh, it's nice to be back on watch.
You were on watch last like.
It was. After a long hiatus.
You were on J archive duties.
That's correct.
Did you sit any Argus last leg, Sam?
I did, actually.
Okay.
For
Nice.
like half an hour.
Nice job.
Did you sit video too?
That was during Simone Clipper, yeah.
Oh, yeah.
Oh, I remember that. That's
I haven't quite caught up to Allison in
Yeah.
sitting every position in the control van, but
She hasn't
getting there.
done nav yet.
That's true.
Really?
I did once. I did a bathroom break for someone. I
Nice
moved
job.
the ship 3 times. It's very exciting.
It is.
Stock granted.
I've never called in a ship move.
What do you think I'm? Well, you know what? We got 3. How many? 3 weeks? We'll
Yeah.
trade. You know, I'll let you sit here. I'll take the hit and fly her. So,
Thanks, Rennie.
Yeah.
Real gentleman. I've done the DP screen from up on the bridge. I've moved the ship.
Yeah.
That was fun.
Anemone.
When we switch to the new system, it's gonna take a while for me to figure out, to get accustomed to it. I'm so used to staring at
You're
this
not on
thing.
talking. There you go.
Time to zoom on that anemone.
Anemone zoom eye. Do I need more jam? Or I need more thruster? There we go. Okay, Ed, you can zoom, please.
Coming in.
A little bit of current down here. You can actually see it in the
Yeah,
Holding.
anemones.
a lot of
Angles.
sand ripples. And yeah, he's moving around. There's that thrusters.
Sand ripple, bullock.
Another of those little bubble
Coming in a little
sponge or tunicate there. Can't tell.
What was that? Just below it?
Yeah. Another one of those little bubble ones. There it is.
Yeah. So bad. It's brilliant.
Okay, come wide, please.
Good look.
Coming
All
wide.
right.
Bridge nav. One more step. Thank you.
Full wide.
I didn't catch that one in time. Hopefully no retrograde. Fingers crossed.
Oh, we just, yeah. There we go.
Looks a bit steep here.
Yeah.
Indeed, we still have that wall in front of us.
Am I seeing retrograde action?
Could be temporary retrograde, hopefully not permanent.
What is permanence?
That's a good question.
But actually, like, I don't know what you mean. I don't know what you mean in this case.
Sometimes it does a little loop versus like a, hey, we're just going to continue to go.
Yeah. Until you stop it and start it again.
Yeah.
Okay.
But to answer your other question, nothing is permanent. You can never go home again.
You can never go home?
You can never go home again.
Why?
It's not the same. It's different. You know, everything's
Wow.
changed.
It's where your heart is.
Yeah.
Huh.
You're
Home
always
is
there.
in front of my lungs.
Hmm.
Do we want to zoom on this coral?
Oh, yeah.
Okay, go ahead, Ed.
Partial.
We get a little polyp zoom, maybe?
Or something like that.
Oh, that's spicy
Yeah.
zoom. Yeah.
There it is.
It's cool.
You're creeping in.
That looks like an octocoral.
There's a big fuzzy one.
All right, come on.
Shaggy.
Zonkers.
But it's not a full retro, I don't think.
Come back in just a punch.
Just kind of waiting it
That's
out.
steep,
3
huh?
beams.
3 beams.
What were the beams we were losing last leg?
1 and 3.
Okay, we lost 4 right now.
Hmm.
Let me turn my head, see what happens.
Got him back.
Funny that.
Holothorian.
Is that
Sorry,
a
it was another one.
encrusting sponge or a tunicate
I
or
don't know,
something?
yeah. Let's check it out.
Sponge, I think.
Yeah.
Definitely
Sponge.
spongy.
Okay, zoom in, please.
Nice.
Very spongy, yeah.
Crinoid there too.
Hmm. Yes, quite.
Okay. Come
Could be a demo sponge.
That ship move is now moving. I will call in the other one sooner this time.
Roger, thank you.
How do you date a sponge?
Take it
Swipe
to
right.
dinner.
Oh, what do we have here?
Swipe right.
Is that another
There's another
mushroom?
mushroom
Little
coral.
hanging fruit there.
Have we talked about the geology at all here? It looks kind of basalty,
We have a,
pillowy.
got a nice sample right off the bat.
Yeah, nice.
Didn't think it would be that easy, but yeah, it's heavily encrusted.
Are we calling some of these pillows?
We have not.
Okay. I just saw a couple that look like
Let's
they
switch it up a
collapsed
bit.
on
Let's
top.
keep it between
Yeah, it's
17
hard
and
to-
22 now, with like a 19 meter.
A couple that look like
middle.
what?
They had maybe
So a little lower down. Sound good?
been
Okay,
pillows
cool.
and then collapsed.
Oh,
The top collapsed, kind of roof collapsed in.
Bridge now.
like a bubble.
We'll add one more step to this. Thank you.
So
Yeah,
keeping
this
the
looks pillowy.
keeping the vehicles higher apart, a little more conservative, but you get a
What
little
would have
less
made them pillows?
flexibility, a little less
I'm seeing more flow
ability
than
to
pillows.
zoom
but
in on stuff. Coming down lower is a
I'm
little
not an
more. You gotta
official
watch it a little more,
geologist.
but you
Play
get
one
better
on TV.
handling of the vehicle and more room to play. So yeah, 19 centered up, but 17 to 21 or so on either end.
Yep.
Alright.
It's like a holothurian there to the left of the lasers. Purple.
Yeah. Hiding.
Oh, and to the right, actually a red one.
Oh yeah, nice.
Mm.
Are we looking for zooms on holothurians? Is that a thing we're after? Just want to observe them.
No,
Okay. Emil doesn't care about holothurians.
nah.
Uh-huh. Hey,
Hey, this
look
looks
at that.
neat.
Is that a
Hey,
coral
Ooh.
with
that
a
is nice.
brittle star?
Nice.
Guessing.
They're doing this. Oh yeah, brittle star on it. Quite wrapped up.
Okay, Ed, go ahead and zoom, please.
Huh.
Is that a Paragorgia?
Definitely bright pink. Sarah's saying that it's moving a little bit, which makes her think it's a bubblegum.
Yeah.
It's very thin, though.
Is that just one sea star associated, or a few?
It's a brittle star, and it does appear to be one.
Huh.
Can't tell. I don't know. It
Maybe
is
not,
kind of,
yeah. I just think it's just one.
yeah, it's just really long legs and not enough coral.
Ooh, I'm losing it. Sorry.
Yeah, it could be.
It's hard to say.
All right, come wide, please.
Wide eye.
He's really fond of that coral.
Yeah, I'm gonna hold off on another move there just as we move up and out.
It's bright pink, too.
Small
Roger
approaches,
that.
and once we get up a bit, I'll call another one in.
Oh, yeah.
Roger. Come up in your delta, please.
Oh,
Is that a fish?
That next target might be a good spot for watch turnover.
watch turnover. What time is it?
11:22:27.
Wow, time flies when you're doing stuff.
Collecting. Some good samples.
Is that, it's just a C-pen.
Oh, where's the C-pen? Right
To the
there?
left there.
We do want to zoom on C-pens. Yeah.
Okay.
I don't see it.
Sorry, right there next to the anemone to the
Yeah.
right? Yeah, right there.
Oh, I see it. Yep.
Good eye. Oh, there's a bunch of stuff going on underneath that rock.
Okay, Ed, you zoom on the C-pen,
Yeah.
please. Got more zoom. Ooh.
Full zoom.
Oh. Bouncy, bouncy,
All right.
bouncy. Okay, come wide.
Right.
Thanks.
Thank you.
How about a halipterus for that one? Could be. Go C-pen for now.
Halipterus. Oh, T-Top C, not that far away.
All the polyps were on one side, right? Versus being
I thought it
fanned
was.
out.
I didn't see them all on one side. Oh, is that a Chrysogorgia or
Oh.
something?
Ooh, that's pretty.
Maybe, yeah.
I can't tell. We're too far away.
Is that a precious coral?
Hard to see what's going on there.
Okay, Ed, zoom in, please.
A lot of little shrimps and squat lobsters living in it.
Yeah.
That is bushy.
Was that you making that sound?
Yeah.
Picking up the Hannafordism?
Wow.
Looks kind of
Yeah, I
like
did pick that up, didn't
Chrysogorgia.
I?
It looks like Chrysogorgia.
Yeah,
It does.
I think. But,
small.
Yeah, it's not very big. There's a crinoid in there, and some shrimps.
Okay, come wide,
It's
please.
hard to see the skeleton,
Yeah.
but just the polyps from a distance.
You come up in your delta, please.
Yeah, we have concurrence. Chrysogorgia from ashore.
Perfect.
Cool.
Look at that beautiful one here.
Man, I sailed enough with Steve
Ooh, that's a pretty sponge.
to
Ooh, pretty.
identify a lot of these things. I feel
Looks pretty
it
big.
speaks to his encyclopedic knowledge that
Okay, zoom in, please.
It's
I'm
the
gonna try
same
to
one
redeem
we saw
myself
earlier.
by spinning
Does it
around
have
this
that
and coming up
cap
it.
on it?
Oh, that'd be cool.
Yeah, I think so.
Nice.
Yeah.
Oh yeah, gorgeous. So that's the
But no prisoners.
No prisoners.
euplectellid.
No. All right, come on.
I'll just do that one. That one.
Okay, I think we get a ship move in.
Okay. Bridge nav.
Whoa. Getting some good boulders now.
Yeah.
One more step, 30 meters, bearing 055. Thank you.
So we left Kiribati now, the Republic of Kiribati?
Yes, we're
Yeah.
in US easy,
Oh, of course.
right?
I'll throw another sea pen down. Yes, we are.
Yes.
What's the pink thing on the rock there? Cucumber?
Hawthorne, yeah.
Yeah.
Oh, there you go.
Another sea pen down below.
Yeah, I might be able to tuck in there and line it up.
Cool.
See what I can do
It's
for
cool, cool Argus shot right now with the lights, kind
Yeah.
of like
Yeah.
panning down the terraces of these boulders.
you. There's a. Oh, never mind. Just kidding.
Okay, another shot for the sea pen here. Getting out the pages.
A little bit of current here.
It's
Yeah.
localized right here behind the boulder.
Yeah. Oh no.
Oh, this one does look like they're all on one side.
Yeah. Oh yeah, it does look like they're on one side. Okay.
I mean, either, you know, either side, but.
Oh, it's hard to tell, huh?
Yeah, you're right. Can't tell till we get. We might just be blowing it around.
Yeah, it's down slope for sure. Can we get a zoom on that, please? Before I lose it all. Terrible.
Yeah,
That's one side.
one
Yeah.
side. Yeah,
Okay.
for sure.
How about Halipterus then?
Okay, come on. That was awful.
Does that one look like the last one?
I don't
Hard to
remember
say.
it well enough.
Yeah.
Because there's another one that's like that, but it's red, and that definitely wasn't a red stock.
More sediment.
Okay. Yes, this one looks like Halipterus.
Yeah.
Maybe not the last one. Okay. 50-50 here.
Yeah.
Yeah.
Oh yeah, that's a good shot.
Yeah, a really good shot of this sediment.
Sorry, I was looking over
I'm just teasing you.
Halipterus.
They're doing it again where they're looking at other non-live pictures of the ocean.
This is a live stream.
You
The
can't
bottom
get
of the
any
ocean.
more live than this, unless you're in Alvin. And then it'll be like a millisecond faster or something.
What's over to the right, Herc? Is that a boulder?
Boulder, Colorado. Hey, there's a sea pen. Let's look at that.
Oh yeah.
And then we'll check out the boulder. Okay, Ed, zoom in, please.
What am I zooming on here? The C-SPAN?
C-band, yeah.
Yeah.
The sediment.
Yeah,
Yeah, I'd say it's the same.
could you keep all the sediment in view? Thanks.
Yeah.
Kind of a different color, but
A little redder.
This does look redder.
Oh, now I'm touching the ground. Ay, ay, ay.
Are you trying to pirouette, or
I'm
you are
just trying
pirouetting?
to not be bouncy, and I'm not doing a great job of it today.
Yeah, that's the whole thing.
There you
What's
go.
wrong with that?
That's
Very
fine.
nice.
Gorgeous. Nice job.
Mm.
Looks like the current's picked up a bit.
Let's go check out that boulder.
Yeah.
There's a little bit of current here, yeah.
That one.
There's your sediment shot.
Beautiful.
Suitable for framing.
Hmm. Looks like it, because you can see the tethers whipped out
Rocks
to
on your right.
the
It's
right.
not. It's definitely non-zero.
Yeah.
It's not
It's a
extreme. but
non-zero current.
It's non-zero current. Yep.
That ship move has ended.
Okay. Want to keep them going?
Yeah.
That depends. Yeah, if you want
Yep,
to
just
keep
keep
going.
going.
To speed up. Bridge nav. One more step. Thank you.
What was the bearing again? 055?
055.
Thank you.
So
So one of our viewers is inquiring about what we do with the samples once they go up to the surface. So the biological samples go to our wet lab, correct? They're processed and made available for scientists on shore.
Yes, and all the biological samples will go to the Harvard Museum of Comparative Zoology, and the geological samples will be sent to to URI at the Marine Geological Samples Laboratory.
Come up in your delta, please.
Thank you.
And for folks watching on Nautilus Live, if you watch to the end of this dive, which is another 20 hours from now, you can see when the vehicles come back on deck, all the science team, all excited with buckets going out to collect samples, and then our cameras switch over to the wet
I
lab.
want to see a
So
number lower than 16.
you too can see how samples are processed.
19
see
is a
Gina
goal,
there.
But yeah, it's like 19, 22 to 17 is a good number, but 16 is probably a good lowest.
And Gina, you've been
Right
keeping
now.
track of all of our samples, right?
Yes, we have a total of 3.
So what
As
kind
of
of
now.
information are you collecting?
So I'm kind of notating where the samples are being located on the ROV, recording their number as well as their in situ environments,
Yeah, that looks good.
and if there's any associates with the sample. So far we have an acorn
So
worm,
Try to keep 22 as the max.
basalt
17
rock,
is the minimum.
and a coral snip.
Or 16 is the minimum or something like that.
Cool. Those are going to be interesting to see when they come back up.
So once they come back up and they go into the lab, what type of data do we get from them? Do we measure them, take pictures?
All of the above. Yeah. The form that Gina was just talking about also includes a wet lab description. So you have both the in situ, when we've seen it on the seafloor description. When it comes back up, it gets another description. Sometimes it looks a little different, but also once you have it up close, you're able to see different features. It's measured, photos are taken, and then sometimes subsamples are taken. So if the coral comes up with associates, each subsample or each organism is taken off and subsampled. And sometimes even from there, subsamples are taken of those individual organisms. So like with the coral, sometimes it will be split into several pieces for each researcher that we're working with or institute we're working with. And all of that is very well tracked and tagged with sample numbers to make sure we know where things are going and where they came from.
Awesome. Thank you, Sam.
There's a good current shot.
Yeah. It's a pass shot now. Bridge nav. We can add one more step. Thank you.
So is the top of this little hill, that target, is that 1 kilometer from the start?
No, but we also had
We had launch, I guess.
we had water, yeah. It is still pretty good though, 600.
That's not bad.
So I think a kilometer is definitely reachable for
Yeah,
everybody.
we made good progress.
Yeah. Now is that just to get to the top or to cross the top?
To get to the top.
To get to the top. So yeah, we got some in the bank. We can cross the top if we. If
I
the
lost
watches
your
are
audio
moving good.
somewhere, Emil.
Say again?
Well, there, that's better. I just couldn't hear you.
My finger might have slipped off. I got there, sponge, yeah. That's a pretty neat shot from Argus.
Yeah, really cool.
Looks like a crinoid.
Yeah.
Yeah, we've seen several of those yellow crinoids.
Nice looking coral in there.
Bridge nav.
Ooh, there's a nice coral.
We'll add one more.
Beautiful.
Thank you.
That looks also like a Paragorgia. Can we
Mm.
zoom in on that one, please?
Yeah.
Really nice.
There's also quite a large tunicate next to it. Wow.
It is, yeah.
Over just about 10 centimeters. Whew.
Wow.
Wow, cool.
It looks like it's in the exact right position in that current. To
Yeah,
just
The coral has got like a donut shape.
it's gorgeous.
It's like a
Yeah.
big frosted
something flew by and took it all out of
That
it.
doesn't have that quite vivid color for the Paragorgia.
That's
I wonder
true.
if it's
not
that
as-
other one.
It's more pale, yeah.
And
Okay,
it's
come
stiffer.
on.
It's not moving. And we were told that if they're not moving, they may be.
Come wide, please, Ed. Thank you.
They may be.
Corallidae?
Corallidae, hemicorallium. Perhaps?
Yeah, maybe.
Perhaps, perhaps, perhaps. Confirmed by Tina.
Yeah. So it does kind of level out a bit here. It's really good bathymetry data, I'll tell you.
Yeah.
Is that ours from today?
Yeah.
Yeah. Awesome.
This is a cool fracture here.
Yeah.
Yeah.
There's some stuff going on right just above the lasers. What am I seeing there? Some yellowness.
Yeah.
Ooh.
Can we zoom in there, please?
What is that?
Those hydroids or are those coral?
Oh.
Those look like coral, don't they? Look like polyps.
There's something red on the bottom right,
Huh.
too.
I have
What
lots
are
more sound once
those?
you get
Okay,
sorted.
go ahead. I think I'm stable.
That is exceptionally interesting.
Wow.
Oh.
Those are
One,
coral,
two.
but they're like
Yeah, they're
There's a
octocoral.
bunch of them.
they're a
Wow.
lot. They're like connected.
Are they connected? Are they actually weaving through the rocks there?
Looks like they're connected.
These are
Each polyp.
That's wild.
Those
That's
aren't
really cool.
standalone. What is that?
Stolonifera?
Oh, yeah. Hold on. Yeah.
See if I can
I'm
look a
trying
little
here.
bit to the right
I've
here.
never seen that before.
That's
Neither have I.
cool. Oh, and they're all connected, yeah.
Looks like what do they call that in plants? Rhizomes.
Yeah, that's beautiful.
It's very funky.
Yeah.
But they're all
Really
connected
cool.
like that.
Yeah, I think Emil is right about the stoloniferans.
Looks like there's maybe an
Tina's
ophryoid
right
up
about it.
hiding behind
Oh,
that
yeah.
thing.
Never seen yellow.
They really are connected.
Yeah,
aren't
they're
they?
and
Is
they're
this-
Is it possible to sample here? Is this too
Uh,
tricky?
We just got to figure out how we would sample.
I don't know how we
without-
would sample that. Come wide, please, Ed.
Yeah.
Oh, we'd have to step the ship backwards one, too. I
Yeah,
think.
that's fine.
Wow.
How about up on the rock there? Looks like there's some more
Yeah, I see
up
that.
top. That might be a bit easier to get if.
Yeah, there's more there, yeah.
That red thing down below must have been an octopus.
Who do you think it was?
Oh, she's
Really? Or just you're
The
just saying
shrimp
that
or
'cause
an
now it's
octopus?
gone?
I don't know. It looked tucked under the rock.
Yeah.
Yeah, Emil, I don't know how we'd sample that. We'd have
Yeah.
to kind of scrape it out with the tip of the jaws. I mean, we could try that, but it might be more destructive than useful.
How about a slurp?
You
You
still have
think
to break it somehow.
You have to get it off the rock, I think, because they can handle the fast current, so I don't know if the slurp would do
Yeah.
anything.
It was a pretty tricky location.
Pretty hard mode, yeah.
Yeah.
Okay.
Can we get you a sample of that in 16 minutes?
Yeah, exactly. I mean, I'm up to try, absolutely. But,
It seems unlikely.
yeah. So the crack's still down to my right here. Maybe I'll drop back down and check it out again, see if we can find more.
Yeah. Yeah,
But-
if we don't see any more here.
We do have, so what do we have? We have the tips of the jaws are quite pointy, so we could try a little scrape,
Uh-huh.
but
Scrape and slurp. Or
yeah, I don't know how we'd pick it up after scraping. 'Cause there is some current here, so if we
Yeah.
don't immediately slurp, it'll be washed away.
be gone.
I think it'll just be all smashed up.
Yeah,
I don't.
the scrape
It's
smash
like-
and slurp.
Yeah, like the way they were connected was so like one against the rock.
Yeah.
I don't
They
know how
found the
you-
groove in the rock and kind of
Yeah.
followed it along, yeah.
Yeah.
I don't know how you do it. You might have to find a loose rock that has them on it, but that's
Exactly.
Gonna be,
Yeah.
that's going to be a diamond.
That's really cool.
What is that word? Needle in a haystack.
Both of these are really
It's
cool.
not a dime a dozen.
Not a diamond, doesn't. Diamond in the rough.
Diamond in the haystack.
Needle in the rough.
There's probably really cool stuff all along this fracture here.
This is the zone.
Oh, neat.
There is certainly interest from ashore if we're able to get one.
I just don't think it was.
Let's keep
But
our
that
eyes
was the
peeled.
recommendation is, yeah, maybe a rock that's loose.
Yeah.
Let's zoom in on this little